Good morning, my dear students. We will be discussing on mitochondria, chloroplasts, ribosomes, and nucleus. First, let us take up mitochondria. They are glandular or filamentous organelles present in the cytoplasm of animal and plant cells. They are known as powerhouses of the cells as they are the main site for the generation of high-energy phosphate bonds ATP through oxidative phosphorylation. Almut first observed them as bioflats at the end of 19th century, but Benda in 1897 called these structures as mitochondria. The shape, size, and number of mitochondria vary in different cells. The shape of mitochondrion in general is filamentous or granular, but during certain functional stages, other derived forms may be observed. The size of mitochondria varies with diameter ranging from 0.2 to 1 micrometer and length from 2 to 10 micrometer. Mitochondria are concentrated near the spindle during mitosis, but they are distributed appropriately in equal proportion between the daughter cells after the cell division. A normal cell may contain 200 to 300 mitochondria, but its number may vary considerably from a few to 1,000 or more depending on the cell type and condition. Mitochondrion consists of an outer and an inner membrane with a thickness of 60 angstrom. The outer and inner membranes are separated from each other by a space of 80 to 100 angstrom. The inner membrane forms many finger-like projections or infoldings which are called mitochondrial crests or cristae. This inner membrane divides the mitochondria into two chambers or compartments. The outer membrane existing between the two membranes and in the core of the crest, and the inner membrane which is filled with a relatively dense proteinaceous material usually called the mitochondrial matrix. The matrix is usually homogeneous, but in some cases it may contain a finely filamentous material or small, highly dense granules containing phospholipids. Small ribosomes and naked circular DNA are present in the matrix. The mitochondrial matrix also contains soluble enzymes of the Krebs cycles, RNA, and other components for protein synthesis in the mitochondrion. The exterior of the outer membrane is made up of small stalkless spheres packed closely together to give a rough appearance of the mitochondrial surface. The inner membrane, on the other hand, has tennis racket-like particles of 70 to 100 angstrom on its surface. These structures are called elementary particles or F1 particles. Each particle has a stalk of about 50 angstrom long and 30 angstrom wide and a head of about 30 angstrom in diameter. The entire length of the structure is about 160 angstrom. The F1 particles are regularly spaced at interval of 100 angstrom on the surface of the inner membrane. The F1 particles are previously thought to contain all the enzymes of electron transport chain and are therefore called electron transport particles. Recker in 1967 had shown that these particles contain ATPase and were involved in the process of oxidative phosphorylation during respiration. There are about 10 to the power 4 to 10 to the power 5 elementary particles present in a mitochondrion, and the presence of this particle on the matrix site confers to the inner membrane a characteristic asymmetry that is of fundamental importance to its function. The mitochondria have been shown to possess the ability to catalyze the reactions of the Krebs cycle using pyruvic acid as fuel. The pyruvic acid is completely oxidized to carbon dioxide and water with the release of energy.